Good afternoon, students. I hope you are staying safe and have read the videos and the PPT presentation which I have sent you about microbial interactions. Those videos were quite easy to understand, and the PPT has given you the idea of about all types of microbial interaction which takes place. So in this row, today we'll read about the biofilms and the quorum sensing. Biofilm is another kind of microbial interaction which takes place. And the phenomena which is responsible for formation of biofilm is known as quorum sensing. Actually, quorum sensing is just not responsible for making the biofilms. It is responsible for number of microbial interactions, and how it takes place we'll learn later. But initially, we'll start with the biofilms, and then we'll come to the quorum sensing. So, what are biofilms? Biofilms are just a minute. Yeah, biofilms the are formed by microbes, and biofilms are the group of microbes when come together in a cluster is known as biofilms. And these biofilms are present everywhere. Like if you see in this diagram, there are teeth scraps, tooth scraps, and you know that first microbes actually when Anthony Van Leeuwenhoek saw it also found these microbes in the teeth, tooth scraps. So the biofilms are the first which were seen actually in the microscopes, and then they are found in pipelines, sewage treatment plants, sewages, and so they are found everywhere where liquid and solid mix together. And the other requirements are that a nutrient should be there, a surface should be there, and the microbes are there. So basically, biofilms can be formed anywhere where the solid and liquid meets. I've given you a number of examples like the riverbeds, shower floors, hospitals, oil, water and sewage pipelines, sink drains, inner ear, teeth, plant, soil, cow rumen, lung, and gut. And these by the speciality of the microbe, these biofilms are that these biofilms are very resistant to number of antibiotics because they secrete number of chemicals or number of compounds which can help them to get rid of antibiotics. So biofilms are more virulent than the single microbe. Secondly, these biofilms are formed in different niches, but there are some common attributes. Like they need a surface for the attachment. Mode of growth for all of them is similar, that initially they required a signal to form a biofilm, and then the biofilm grows, and when the biofilm grows up to a size, then some of the bacteria will separate from this and forms another biofilm. And their ability to spread, how they are nourished, how they maintain themselves as a colony, all of these are the common attributes which are found in biofilms. The only requirement for the formation of biofilm, as I told you, is microbes, moisture, nutrient, and the surface. One more important things which we need to know is that microbes, the biofilms can be formed by a single microbes or the mixed species. So biofilms are communities of microbes that live on surface in a cell-producing matrix of extracellular polymeric substance. Basically, these biofilms, these microbes, when come together, they secrete a slime-like compound known as extracellular polymeric substance. And this extracellular polymeric substance consists of number of chemicals like the proteins, carbohydrates, polysaccharides, DNA, and lipids. And there are several functions which EPS does, is like it is required for the addition to the surface so that the biofilm can grow, and the cohesion and aggregation of the biofilm members. These biofilm members could be interspecies or the interspecies. Cell remains in close proximity to the intercellular interaction. DNA exchange, although this has not been proved, but some of the scientists or we think that the species which are present in the biofilm, they also do some of the DNA exchanges like a conjugation kind of thing, so that they can be more rigid to their environment, more adapted to their environment. And then the water retention is one of the major function of APS matrix because the biofilm should not get dried. It is a protective barrier against number of antibiotics, as I told you. and it also absorbs organic and inorganic nutrients for the biofilm members. EPS form up to 90% of the dry weight of the biofilm, so it's a huge amount. EPS produced by biofilm organisms themselves. So the micro which are present in the biofilm, they self produce the EPS. And biofilms can have all kind of textures like a fluffy, smooth, roughness, and filamentous. So these are all these kind of appearance are also found in number of microbial colonies, as well as they are the same attributes which are found in biofilms. EPS matrix consists of hydrated biopolymer including polysaccharides, proteins, DNA, and lipids, and then the water. Water helps in, it is highly hydrated, helps prevent cell desiccation. Polysaccharides, there are two types of polysaccharide which are present in EPS. Homopolysaccharides like sucrose derived glycans. Homopolysaccharide means that they are made up of same kind of monomers, and the heteropolysaccharide which are made up of different kind of monomers. So homopolysaccharides are mostly sucrose derived glucans and fructans, while the heteropolysaccharide are neutral or charged sugar residues. EPS matrix of Pseudomonas aeruginosa have three major polysaccharides. This is just an example, like the alginates, linked uronic acids. So these are the uronic acid moieties which are linked together, and then the PEL, or known as pellicule, which is glucose-rich polysaccharide, and PSL, repeating unit of D-mannose, D-glucose, and L-ramenose. All these polysaccharides are required for initial biofilm establishment and the maintenance of the mature biofilm. So, which means these all polysaccharides are required right from the origin of the biofilm till its maturity. Then the second is proteins. So proteins are generally, as we know, that number of enzymes are required for degradation of different biopolymers which are present in the biofilm or which are trapped in the biofilm, so that these can be utilized by the microbes as a nutrient. So number of extracellular enzymes are present that can degrade biopolymers. Then there are structural proteins which can maintain the structure of the biofilms, and these are non-enzymatic proteins like lactans, which can bind to carbohydrate and help to stabilize polysaccharide and help link surface with EPS. And then there are motility proteins like flagella and pile can also act as a structural protein in the biofilm matrix, because in the biofilm motility proteins are not required because the microbes doesn't move, they stay at one place. So these proteins can also act as a structural protein to maintain the structure of the biofilm matrix. However, when the biofilm is mature enough and then it is required to form a new biofilm, these motility proteins are again active, and these flagella and pili, they with the help of this flagella, the bacteria, the microbes moves to another place, some of the microbes, and they start forming a new biofilm. Then there is an extracellular DNA which is also present in the EPS matrix, and this could be present due to the lyse cells, the cells which are dead and their DNA remains in the biofilm, can function as a structural binding component. DNA transfer between cells, as I told you, it's not known, but we think that number of microbes do different microbial phenomena like conjugation, so that this DNA is transferred from one cell to another and makes biofilm members more adapted to their environment. And the lipids which are present are lipopolysaccharide can function in addition of biofilm to surfaces, and the another one are biosurfactants like Pseudomonas aeruginosa. The hemolipid can function in initial microcolony formation and the cell detachment. So cell detachment is needed when the new biofilm is going to take place. New biofilm formation will going to take place, right? So I guess you have understand this. This is a micrograph of biofilm from the Nature Reviews of Microbiology in 2010. If you want, you can go through this article and you can find more about these compositions. This is a diagram from the same article of 2010. Different microbial biofilm, their formation, their constituents, and how they are in how EPS is connected as a structural thing for the biofilm. And then this is a alginate, which I told you in the Pseudomonas aeruginosa biofilm, and then the lipases which are required. So all of these has been, these are the models, and this is a electromicrograph from the same paper. Model of biofilm formation. So this is from a article in an article from 2005, which shows the formation of the biofilm, how the formation of biofilm takes place. So initially, some of these planktonic bacteria, they come and adsorb to a surface, and they, as soon as they attach to a surface, they will lose their pili and the flagella, and the attachment will become irreversible. As soon as this attachment will become irreversible, they will start sending, secreting some signal molecules. Remember quorum sensing, which I was telling you. We'll discuss about this later. And then with this signal molecules, more microorganisms come together and they start forming this biofilm bigger. After some time, this biofilm will become larger in size. This is an extra polymeric substance line which I was talking to you, which is covering this all bacteria together. So this is a mature microbial formation, and when this mature microbial formation, this biofilm become mature, what happens? Some of these bacteria, these will detach from this mature biofilm and again start the same process. So this is a process for the formation of a biofilm, right? So this is again the biofilm structure from the same paper. This shows how the nutrients input takes place in the biofilm and how they are resistant to the antibiotics. So basically, on the surface of the biofilm, the mostly negatively charged particles are there, compounds are there. And these negatively charged compounds, what they do, they immobilize the positively charged antimicrobials right on the surface so that they doesn't reach to the microbes which are present. Secondly, the oxygen gradient is there. As the biofilm becomes dense, the oxygen gradient becomes low. So it is higher at the surface and it is low in the middle of the biofilm. So fast growers, which need more oxygens, are growing near to the surface, while the slow growers are growing inside. So you can see that, right? Similarly, the nutrients higher nutrients are found near the surface. So fast growers need more nutrients, so they are growing near the surface, and while the slow growers are growing inside. So this is a very good model for inter-species coexistence in the biofilm. So that and all these species are benefited by one another in one or some another form, right? So next, so what is the signal for biofilm formation? So that is I was talking to you about, that attachment to a surface is likely a signal for expression of biofilm specific genes under control by quorum sensing. So biofilm traits under quorum sensing control are EPS production, biosurfactant synthesis, and swarming, right? So these are the traits which are controlled by the quorum sensing. So quorum sensing is a phenomena which induce number of microbial interaction, and one of them is biofilm formation. So quorum sensing is a mechanism by which bacteria assess their population density, resulting in collective gene expression. And this collective gene expression is responsible for number of microbial interactions. Means of cell to cell, between cells communication, involved in the production and detection of extracellular signal molecules called autoinducers. Autoinducers freely diffuse across membranes in either direction. They can go inside the cell and they can come outside the cell. So these then freely diffuse autoinducers. So what are autoinducers? Autoinducer, you can say these are the bacterial language. Bacteria talk to each other with the help of autoinducers. And the very famous autoinducers which are found in bacteria, typically in bacteria, are known as homoserine lactones. So they are found in most of the gram-negative bacteria, also in some gram-positive bacteria, but mostly in the gram-negative bacteria. And those are known as homoserine lactones as autoinducer, although others have been also been identified. In gram-negative bacteria, there are short peptides which are present, which act as autoinducers. A universal autoinducer, or also known as autoinducer 2, made by both gram-positive and gram-negative, and used for interspecies communication. So autoinducer 2 is a universal autoinducer. We call it as AI2. So autoinducers are acyl homoserine lactones. As I told you, this is a structure of acyl homoserine lactones, most common. Autoinducer 2 is a universal signal molecule. As I told you, this is a universal signal molecule, also known as AI2. And other non-AHL autoinducers, which I told you, are mostly could be peptides, some of the peptides. So these are some non-AHL inducers. They are responsible for virulence and EPS production in Pseudomonas species. Example of a specific acyl homoserine lactone produced by gram-negative bacteria. So these are some of the different homoserine lactones, like the C4 homoserine lactones, C8 homoserine lactones, 3-oxo C6 homoserine lactones. These are in different bacteria. You can see this is in C4HSL is in Pseudomonas aeruginosa, C8HSL is in Vibrio fischeri, 3-oxo C6 HSL is in Vibrio fischeri, 3-oxo C12 HSL is in Pseudomonas aeruginosa, and 3-hydroxy C4 HSL is in Vibrio harvey. Right? Got it? So now how it works. How does quorum sensing work? Cells capable of quorum sensing express autoinducer at a basal level. Autoinducer only if many of the same cells are near, cells of same species producing the same autoinducer. When cell number are high, 10 raised to 7 to 10 raised to 9, when cells reach a quorum, autoinducer concentration will be high. When autoinducer concentration are present at a certain high level, they can bind to an activator, trigger gene expression. So this is the phenomena, right? You understand that initially cell always secretes some autoinducer at the basal level, but that is not a quorum sensing. They keep secreting it, right? But when the cells, when the cell density is high, this autoinducer is secreted by the yeast cell, and then the concentration of the autoinducers increase. And when it reaches to a certain level, certain threshold, it will trigger the operons, which will help trigger different gene expressions, right? So this is a phenomena. So classical quorum sensing, it was discovered in 1970 in the bioluminescent bacteria, Vibrio fischeri and Vibrio harvey, marine bacteria that can colonize in squid. There's a very interesting story about it. And I would recommend that you should go through this YouTube link, which is written here, The Secret Social Lives of Bacteria by Bonnie Besler. So what this video actually explains is that how squid, organism which is present in the sea, this this organism has a marine bacteria which is known as Vibrio fischeri, which is growing in its gut. And this organism, what happens? This organism in the daytime it remains in the rocks, creeks near the sea. It remains in the creeks. And in the night, when there's a high tide, this organism, squid, goes in the water to find the food for itself. what happened, because this squid is also a prey for number of fishes and other organisms in the sea. It inhibits itself to get to get attacked by different organisms in the sea. It takes the help of Vibrio fishery, and in the return Vibrio fishery is getting a place where it can reside. So what happened? It is growing in its gut, this Vibrio fishery organism, and when the concentration of this, when the cell density of this Vibrio fishery is more than a level, they are producing an autoinducer. This autoinducer will activate the gene expression of a lux operon. This encodes bioluminescence, luciferin enzyme, and then it started giving light. So in the night, when this organism is in the sea, this light is there in its the light is there in its gut, right, in the neck. So what happened, that when the moonlight comes over this organism, the shadow will be there on the in the down, right? So with this shadow is detected by the organism which want to attack this squid. But what happened, this light which is coming from the vibrio, the bioluminescence, can mask that shadow exactly, so that this organism while this organism move, no other prey can see this squid, and the squid can get its food, right? So this is like a self-defense. Okay, so the light of the moon which is coming on the organism is masked by the light which is produced by the vibrio fishery in the gut of the squid, and in this way it saves itself from the prey to get preyed, and while finding its own food. And as soon as the sun rises, this squid again remain in the creeks, but this bacteria is growing so much in its gut, which is not required in the daytime. So what happened? This squid organism, it vomit out all that bacteria from its gut, from its neck, and only a small amount of cells remains in its neck for the whole day, and they keep growing till the evening, till the night. And by night, these bacteria grow so much that they will start producing the autoinducer again to activate the autoinducer, and this autoinducer will activate in turn the lux operon for the bioluminescence. So this is all things. This is everything which this is a very very good example of the quorum sensing. So in quorum sensing in the vibrio fishery is operated by two operons, OR operon and OL operon, and the autoinducer is 3-oxo C6HSL. So what happened? This OR operon encodes for synthesis lux I that makes this autoinducer 3-oxo 6C-HSL, and this 3-oxo C6HSL, it is detected by the regulatory protein LuxR in the OL operon, and as soon as this is detected by LuxR activate the transcription of lux bioluminescence genes luciferase, so that the light enzyme luciferase will induce light. So these are the two operons. So we can see them in this diagram, right? So initially, you can see that the cell density is very low. There are only 4 or 5 cells. So this autoinducer is there, but it is not in so much concentration that it is detected by LuxR. So the basal transcription of lux genes. And when there's a high cell density, you can see in the background, this autoinducer is everywhere, and it is detected by LuxR, and as soon as it attaches to a LuxR, the LuxR gets out of this operon, and the expression of this lux operon takes place so that the light is produced. So this is the whole system which is taking place in the Vibrio fishery. So there's another YouTube link by Holden et al. In 2000, Quorum Sensing. I recommend you to go through this YouTube link too also. Then there's another example of Lux-Q-Luxo phosphorylase relay in Vibrio harvey. This is from another paper in 2004. This is mostly for the secretion of protease and other genes. And yeah, so this is for phosphorylation. If you want, you can go through this paper and read it. It is also present in the book known as White on page number 560. I can send you that book also. This is another example of the same thing. This is from the same book which I told you, White. Then quorum sensing regulated activities. So quorum sensing regulated activities, there are a number of activities which are done by the quorum sensing. Most important, which I told you, is a biofilm formation. And then there is a bioluminescence, symbiosis, virulence, competence, conjugation, antibiotic production, motility, and sporization. All these activities are actually guided by quorum sensing. So in the biofilm, related traits under quorum sensing control, there are a number of traits which are under the control of quorum sensing, includes surface attachment, extracellular polymer production, and biosurfactant synthesis. And as well as decreased expression of genes needed for motile life. As I told you, that bacteria which is present in the biofilm are not sessile. They are they are sessile, so they live a sessile life. And so the genes required for the expression of flagella or pili doesn't express, their expression decrease, while the increase in the expression of the genes which are needed for sessile life to remain at one place. So in Pseudomonas aeruginosa, to measure AHL-based quorum sensing systems are less, and RHL. Less in the less, the transcription activator is less R, and the autoinducer is 3-oxo-C12-HSL. It was discovered that the glucose-rich EPS spell was under the control of the cell density dependence less system. So initially, if you remember, I told you about Pseudomonas aeruginosa, and I told you about one of the polysaccharide which is present there is spell, and this is under the control of less R, less system, less quorum sensing system. And then another one is RHL system, where the transcription activator is RHLR, and the homoserine lactone is, or the autoinducer is C4HSL, and this is required for the rhamnolipid synthesis under control of this quorum sensing system. Plays an important role in the initial attachment of biofilm and the detachment of the cells. This is a schematic diagram of the same thing which I told you. So initial genes, then there's the activation, signal is produced, it becomes and the active, and then the target genes which get expression, right? So I hope you have understood this. This is from the same paper. All right, so this is all about biofilm and quorum sensing. I hope you have understood this. Try to go through this video again and again so that you can understand this. And we'll discuss the question through Piaja. I hope all of you have done your registration on the Piaja, as well as you can ask me the question directly through the WhatsApp or by email. I'll also send you this PPT in the email, as well as the link for this video. Thank you very much.